Hi, my talk today is titled Spatial Single-Cell Functional Genomics and Cell Decision-Making. My name is Jay Lee. I'm an assistant professor at Cold Spring Harbor Laboratory. We know that various signaling processes and mechanical transduction processes are involved in developmental patterning and developmental signaling. Here I show you an example of Drosophila wing imaginal disc on the upper left corner. For those of you who are not familiar with Drosophila developmental genetics, Drosophila wing imaginal disc has been a prime example of and prime system that has been used over the past several decades to dissect various morphogen gradients and morphogen signaling pathways to study how organ size, shape, and patterning was established prior to them actually forming the future antenna, eye, wing, or the leg. Here, showing the green stripe is DPP secretion gradient. So DPP is a morphogen similar to the TGF-beta family or bone morphogenic protein family that establish a concentration gradient from the green line and spreads out across to the anterior and posterior compartment of the wing. And this actually then translates into discrete zones of self-specification. Now, beyond morphogen gradients, which is classically the patterning cue, we now know that various other cues shown in the lower right corner, including cell adhesion signaling, cell polarity signaling, cell cortical tension signaling, and cell proliferation signaling, they all participate in overlapping with a morphogen signaling to control organ size and shape. And the question is, how do they actually come together and execute and orchestrate this complex process in vivo? Well, we have, after even despite decades of studying this problem, it's not clear how to go about this systematically and comprehensively. So we know that showing in subpanel A, we know that DPP morphogen gradient, which is smoothly spread out across in space, controls discrete zones of gene expression. Here, for example, Brinker and OMB and cell, and shown here. And it's not clear how these discrete zones of gene expression are established from these smooth morphogen gradients. Some things are known, which is that we know that gene enhancers and promoters and their cis-regulatory elements, including repressors and coactivators, can convert analog signals into these binary signals, right? And on-off signals, right, in gene expression. But people have done QTL analysis and other type of functional genomic analysis, and there are clues indicating that beyond just enhancers and promoters, these cis-regulatory elements are spread out across not only these regions, but also across the entire life cycle of RNA, meaning at the capping stage, splicing stage, RNA translocation stage, translation stage, and elements that regulate the RNA turnover. But how one would go about actually mapping these functional elements throughout many different cell types in vivo during different developmental stage is a major challenge, both conceptual and technological. So again, just to reiterate, so cis-regulatory elements and their modules integrate multiple signals. So if you're thinking of morphogen signaling and tension signaling and adhesion signaling all coming together, we cannot just restrict ourselves into thinking that, well, we should just study enhancers and promoters of the gene. But in fact, all the functional regulatory elements that control where the gene is expressed and how they're translated is spread across the entire gene body, both upstream and downstream. So our goal is to really comprehensively identify these cis-regulatory mechanisms of gene expression and gene translation throughout the entire life cycle of RNA. Again, let me emphasize that we're just going to go beyond study of the gene regulation at the promoter enhancer level, but we think that the way RNA is regulated, RNA biology, and their location is critical for some of the early developmental patterning phenomena that you see in vivo. So again, on-off states of all, our goal is to systematically characterize on-off states of all cis-regulatory elements, genome-wide and transcription-wide in single cells in situ, preferably in 3D. And this way we can actually map out all the decision circuits in individual cells throughout development. And our goal is to also use a defined system for mapping these cis-regulatory elements, which is a classically and also well studied Drosophila wing imaginal disk. But before we get there, we want to have a method that could actually do this. So only way what could really do this is do functional genomics in situ in single cells. Or you could do a variety of tricks where you could do single cell functional genomics followed by mapping of individual cells, ex situ or post hoc. But ideally, the best solution is to be able to do functional genomics in situ in single cells. So in 2014, with this goal in mind of eventually getting us to the spatial functional genomics, unbiased spatial functional genomics in vivo. In 2014, we didn't even know whether or not we could sequence RNA in situ, right? So this is a paper that was published in 2014 in Science. I was a postdoc in the Church Lab, and the moniker for this technology was called fluorescent in situ sequencing, or FISSE. Generally, the protocol goes like this. You have a tissue, you permeabilize it, you flow it in your RT primer, and here is shown in panel 1. And this random hexamer primer initiates reverse transcription, during which amino allo UTP is incorporated. This amino allo containing cDNA in panel 2 is then cross-linked to the matrix using BS-PEG, a non-reversible cross-linker, and RNA is then digested. After washing, now you have single-stranded cDNA molecular libraries in situ in single cells, right? Either in fixed cells or in tissue sections. You can circularize this. You can perform intramolecular circularization using circ ligase, which is then followed by 529-based rolling circle application. And the critical step here is the inclusion of amino allo DUTP during RCA. This enables all the cDNA amplicon libraries to be cross-linked to one another using BS-PEG. Again, non-reversible and inert cross-linker. And in panel 6, what you show is that this, and in the end, creates this very stable and very even library of cDNA molecules you can then sequence in situ using harsh conditions, harsh and stringent hybridization and washing conditions in situ. I'm not gonna belabor this point, but back then we were limited to using solid chemistry from ABI, Thermo Fisher, and they no longer exist because they essentially Illumina took over the entire NGS, and a lot of the sequencing chemistry businesses they moved elsewhere. But back then we still had access to solid chemistry, and essentially AGCT bases have different colors. Each applicant has about 1,000 or hundreds to 1,000 repeats of the same cDNA molecule with a adapter sequence here shown in orange, and you hybridize the sequencing primer N, and then you integrate the first two bases, and then you get a picture down in cycle 1. After imaging, you cleave the sequencing primer, and that essentially lengthens your sequencing primer, to which you can ligate it again, image, cleave, ligate, image, cleave, so on. So you can generate up to 35 image in one sequencing run. And in the end, you get an image like this, where the z-axis is essentially different image cycles in a single sequencing run. This is about 35 cycles you're seeing here. And they've been competition aligned using a very basic MATLAB custom script by Evan Doherty in the Church Lab, who was a graduate student back then in the Church Lab. And so this is, again, a single cell culture fibroblast migrating in 2D. And the hazy stuff that you see in the center is a nuclear background. And you can see the leading edge and the tailing edge of the fibroblast much more clearly from the top view. So this was enough for our first publication to show that, hey, we can sequence RNA in situ, and we can do a lot of interesting regional enrichment biological pathways simply based on how the cells look and where the cells are. But there are severe, well, that's too strong. There are a number of limitations that actually prevented even us and the Church Lab and many other people from taking this technology forward. First, the biggest one was that is the fact that because we're doing 3D cell imaging in high resolution, our imaging time was very long, even for one field of view. So for example, in the previous image we showed you, it was about 2 hours per imaging cycle. And if you wanted to do 35 sequencing cycles, including wash and wash, image, cleaving time, all that stuff, that essentially becomes 2 hours plus times 35. It's almost 2 weeks. In fact, if you were to imagine a way to do 500 base in the future, the imaging time alone mandates that you'll spend about half a year, just half a year for a sequencing run. And that's simply not scalable or even doable. And even with all that time, over 2 weeks of imaging, right? And it doesn't matter whether you automate it or not, this is fixed. So, and I mean, ultimately what you get is a short read length. And as you know, short read length that may be enough for just knowing what RNA is, but anything beyond that, for example, looking for novel splicing sites, novel mutate SNPs, and novel transcription start site, and also skipping over repetitive regions, all those things are not possible with short read lengths. So that was another Achilles heel of this previous technology. And also, we weren't quite able to get our poly-DT primer working in situ, and we weren't sure why. The only thing we really knew could get it working back then was random hexamer, and depending on the cell type, for primary cells you get about 65% ribosomal RNA, but for proliferating cells and cancer cells, you get up to 95 to 99% ribosomal RNA. And that translates to anywhere between 1 to 10 unique mRNA, UMI per cell, which if you have lots and lots of cells, it's okay, but that's about 100 times less UMI per standard single cell analysis, single cell RNA sequencing technologies. So again, so these things all combined together, and finally the killer was that we no longer had access to ABI, solid chemistry, because they no longer sold it. So all these things kind of made the whole technology sound potentially development of this technology very challenging up to about 3 years ago. So comparing NGS to cell-based in situ sequencing, I went over it again. Both are done on same size cover slide or glass slide. In the Illumina NGS case, you extract the DNA or RNA, you make the sequencing library X-Situ in solution, and you put it back on the NGS flow cell where you have a localized PCR reaction to generate these clusters, and you use fast scanning epifluorescence microscopy to generate long sequencing reads, or many imaging cycles up to 300. In PhysIC, in 2014, we have cells position on the slide. We actually make the sequencing library inside the cell while the cells are still attached to the glass. And then we use confocal microscopy and up to 200 Z slices in 3D. And we use anywhere between 20x to 100x for imaging. So conceptually, it's conceptually both can use any type of sequencing chemistry that's available on the market, but really the problem was this. Sequencing throughput was fundamentally limited by the imaging resolution and area size. What do I mean by that? Well, if you look at how this scales, and so you have to be able to individually see these clusters, right? That represent a DNA sequence, right? Or cDNA sequence in our case. So, but in order to do that, you need higher resolution. Higher resolution demands that we spend more time imaging. So if you simply look at, this is true regardless of whether or not you're looking at Illumina NGS or FISSIG. So as you increase hours of imaging, the number of clusters you have, so this particular graph on the left is for FISSIG. So when I say hours of imaging, we're talking about 3D imaging, right? So if you want to resolve 100,000 amplicons inside of the tissue, it takes about 1.5 hours. But if you wanted to get up to 1 million amplicons, right, or 1 million sequencing depth, then you're talking about over 10 hours of 3D imaging. So why does it take so long? So for those of you who are familiar with 3D microscopy, high-resolution 3D confocal microscopy, so this is a Drosophila retina. Individual spheres that you see are DNA amplicons, and you're seeing about 300,000 amplicons in the Drosophila retina here. And in the center, what you're seeing is a optical stalk that coming out of the retina to the brain. So again, in order to get this kind of image, and also obtain 500 base reads from a tissue section, it could require up to a year of imaging. So what we decided to do is, number one, address the lack of sequencing chemistry that can be used in the spatial transcriptomics field, number one. Two, also provide an avenue so that we can barcode individual molecules super efficiently in situ with a minimal number of imaging cycles possible, right? So barcoding individual molecules in situ without exogenous tagging, or having it so that we can do minimum number of imaging. And that's a key take-home message here. So here I'm showing compressed bidirectional sequencing chemistry. You essentially have a rolling circle amplification copy and the sequencing primer in pink. On the left, you have a cDNA start site. We have a mixture of 3 different probes from IDT, and 2 of the probes are labeled with red and cyan. And each probe is pseudo-degenerate, meaning that instead of having just A, G, C, T having different color, we make it so that K, which represent 2 bases, and R, which represent 2 bases, they're represented by a single color. And we do the same on the other side, on the right side. And this side actually represents, because the molecule is circularized, it represents a cDNA termination site. So there you also have R and K, but you use 2 different colors, right? And C here is dark. So what you have is essentially you can decode individual bases, AGCT, based on the way they are color-coded. What do I mean by that? Again, you have a total of 3 probes from IDT on each side. You have K, which could be A or G. You have R, which could be T or G, right? So these are individual, pseudo-degenerate probes you order from IDT. And you have C probe, which will remain dark. So when you do this, you can see from the color table below here that here's a reverse base call that looks at the cDNA start site. You have the forward base call, which looks at the cDNA stop site, right? So and then you can, just by looking at the colors across 4 separate channels, right? And you can actually determine these two bases from both sides of the sequence primer from one sequence cycle. So this means that you can actually sequence 2 bases in 1 imaging cycle. That doesn't sound like a lot, but when your imaging time is so long, it actually makes a huge difference. So sequencing cluster in 3D versus 2D flow cell versus 3D tissue. So in Illumina, the typical cluster density, so this is a metric that a lot of people looked at early on in the early NGS days, but now end users generally don't care about this because it's so robust. So you were getting about 800 clusters, 800 amplicons per square meter in a high flow cell, high-seq flow cell. In INSTA-seg, for a Drosophila retina shown here, size 0.5 millimeters in diameter by 100 microns in height, and this represents about 200 optical sections in Z. So the cluster density here, what you're getting is about 1 million per cubic millimeters, right? So it's like dramatic, and this is a reason why it takes so much longer to image the biological specimens jam-packed with amplicons in situ, rather than these super nice Illumina high-seq flow cells where all the amplicons are nicely spread apart and you can do high-speed scanning microscopy. So now you know why it takes so long and what the expected amplicon cluster density is. So now what we do is taking our bidirectional compressed sequencing chemistry, and we just cycle it. Here in the lower panel, you see you can see that there are small circle numbers 1, 2, 3, 4, 5, 6. So we essentially, these are imaging cycle numbers. So we interrogate the first set of base, and then we image and cleave. We interrogate the second set of base, and then we image, and then we strip the entire sequencing primer complex off, and then we have a recessed sequencing primer in round 2. We do the same thing, and recessed sequencing primer in number 3. So essentially 2 imaging cycles per sequencing primer, and there's 3 sequencing primers, and that's a total of 6. And because we're sequencing 2 bases, that's 6 interrogation points, 6 imaging cycles. That gives you 12 bases, and we reserve 1 base for air checking. So to compare the base calls from the round 6, round 1 and round 6. And we find that generally, depending on the region and imaging quality, about 5 to 10% of the bases are rejected due to the discordance between base calls. So what can you do with only 6 imaging cycles? So again, just to remind you, this is how our sequencing library is generated. So you're seeing a micro-dissected retina here on the left. It's about 0.5 millimeter per sample. And this, and once these cells are fixed, they're permeabilized, and then we use a truncated version of polarity primer. And we, in our initial work, we use NNV-T polarity 10, but now use NV polarity 10. They don't seem to make much of a difference. And the key thing that we are doing here is that we are actually pre-hybridizing our polarity primers overnight with a high stringency condition, so that we're to maximize both the specificity and sensitivity of the poly-A capture. And we can do this because our RNA is immobilized, and you can be followed by a wash-up wash. And so, after you do that, we do standard reverse transcription using MMLUV, reverse transcriptase. So in the green, you show cDNA start site, and it stops at some point, and we call it cDNA stop. And then once after RT, you get rid of the RNA, and then you circularize so that now your stop and start site are adjacent to your sequencing RT primer with the adapter site shown in green. And when you do RCA, this is repeated up to hundreds to 1,000 times per DNA molecule. So you're just essentially hybridizing your sequencing primer onto this repeated green portion to sequence both the stop site and the start site simultaneously in one imaging cycle, right? It's shown here. So the question is, well, even if you were to get 12 bases in 6 imaging cycles, isn't it just way too short to map your sequence to the genome? Well, if you didn't have any prior expectation of where these cDNAs are coming from, that would be true. But if you can do sequencing of these amplicons and have some idea of how well you perform in terms of making sure that your cDNAs are coming from the 3' UTR or polyadenylated portion, right? And if you have a prior, you can actually use that information to refine your reference map. So shown on the right, what you're seeing is if you have a different number of bases for alignment, so let's take 50 bases, right? If you only have 50 base single and human genome sequence, right? Then only about 70% would map accurately to their target, right? And you would literally have to go beyond 100 to really get to the point where you can get 90% alignment, over 90% alignment on a consistent basis. Now, that changes when you say, okay, so what if we only say that our reads only will map to, or only are coming from 3' UTR of the human genome? When you do that, as soon as you hit about 14 bases, right, you're already up in the regime of 90%, right, alignment, right? And when you actually look at, okay, so what if we do paired and 3' UTR, meaning that these reads are separated by a certain gap size, right? And then again, I'm showing you here that up to 14 bases or 13, anywhere between 12 to 14 bases can get you over 92% accuracy and alignment across the human genome. So this is highly dependent on the prior that you have of, how well, where are your cDNA sequencing results coming from? So in order to really kind of know that, what you can do is essentially you can ELISA the tissue, you can pull down your amplicons using adapter hybridization with bound to the diamide beads, and you can do multiple displaced application to get more products, and then these double-stranded DNA can be prepared into an Illumina NGS library. And so for our work, we used Illumina MySeq Paradigm 300. And for all these reads are then separated into the forward section and reverse section representing the start of the cDNA and end of the cDNA. And these paired sequences are then aligned to one another. And what you're seeing here is all the unique reads that comprise a single amplicon. In the yellow section, you see the cDNA start, and in the cyan section, you see the cDNA stop. And our next generation sequencing experiment revealed that cDNA synthesis are indeed coming from the 3' end, where over only a few genes had reads that come outside the 3' UTR in the coding region. I think it was 2 or 3 genes, but everything else got comprised to about 800 genes. All the reads are restricted to the 3' UTR. And if you actually then look at the 3' UTR region and the types of reads that we're getting, on the right, you can see where all the reads map in the normalized 3' UTR region, 100 being where the polyadenylation is. You have cDNA start site and their predominant peaks, three predominant peaks. cDNA stop sites, you also have 3 predominant peaks, which is expected since every start should have a stop. But interesting, what you also see is that for many of the reads, about 15% of the reads, you have these base incorporation, meaning there's a single base pair in cDNA synthesis whenever there is a cDNA stop site. This is a classic signature of some kind of blocking event inducing premature cDNA termination. These blocking events include RNA protein interactions and RNA secondary structures. So what do we actually detect? So now not only can we actually map efficiently our amplicons to the human reference genome of the 3' UTR, but longer read length for each amplicon enables you to map these reads unambiguously to their target genes. So we simply confirm what we knew from the 2014 work, is that FISSEQ, unlike single-cell RNA-seq or bulk RNA-seq, we enrich for things that are highly, quote-unquote, relevant to the biological and physiological process of the given tissue or cell type. So here I show you, on the left, you have the red dot that comes from unique INSTA-seg reads, that's our technology, and blue, which is a drop-seq UMI account, again from the Drosophila imaginal disk. And if there are some genes that correlate, but there are multiple genes that are only exclusive to INSTA-seg or exclusive to drop-seq. If you look at what these are, so things that are unique to INSTA-seg are things that are involved in synaptic organization, glial cell growth, phosphoreceptor regulation, ion channel, and actin-myosin contractile apparatus. So these are highly relevant to the biology of the retina. If you look at things that are unique to only drop-seq, they're almost every single one are linked to ribosomal or ribosomal regulation or composition or translation regulation. So we had almost an identical finding in the earlier 2014 paper. We thought it was very interesting, but we never had a good explanation for it. So, but now we have preliminary data. We're going to update this in our manuscript, which is currently under revision. So our preliminary shows higher correlation to ribosome profiling to gene expression profiling sequencing methods. So our method somehow enriches for mRNA, we think, that are bound to all these ribosome subunits that are present in situ. Again, gene pathway is enriched in INSTA-seg. And this is essentially, if you were to do this with a single-cell RNA-seq or bulk RNA-seq, you would largely get some undefined translational and cell biological processes, general cell biological processes. And now we're able to do this in culture cells. So Drosophila cells that I'm going to show you later on is about 10 to 100 times smaller than typical mammalian cells. So this is about this is a human astrocyte. It's about 20 microns in diameter. Drosophila cone cells can be as small as 1 micron. So in order to get a good sense of how many unique molecular identifier you can get per cell, for example, drop-seq or slide-seq, right? So they all use mammalian cells that are much bigger than Drosophila cells. So we want to get a fair comparison, right? So we perform INSTA-se in a human cell, human astrocyte, and then we use our method, and this is 100x imaging, to obtain up to anywhere between 4,000 to 10,000 amplicons per cell. And here on the left, you can see you can see left and right, one sequence, then one's not, and you can see that there's a dense cluster of amplicons just around the nuclear periphery. And also you can see on the right that there are various kind of irregular pattern structure both in and outside the nucleus. And higher magnification reveals that even within the nucleus, in the cytoplasm, these amplicons are structured into various different compartments. Again, we're seeing reliably about 46,000 amplicons per cell. So again, we see a dense amplicon density around the where you expect to see rough ER, endoplasmic reticulum. You have a dark spot where you expect nucleolus, right? But you again see on the lower left corner, you see these high speckles or amplicon-rich speckles also in the nucleus as well. So there is a amplicon density heterogeneity both in the nucleus, in the cytoplasm. In the cytoplasm, you see this dotted shadow. So for each cell that has a concave-like nucleus, for every cell, there is this amplicon-depleted region that's approximately the size of half the nucleus. It could be Golgi at this where there are fewer ribosomal subunits. So right now we're actively trying to apply our technology to understand the subcellular organization of RNA, mRNA processing and translation. So again, in this image is not just for, it's not visual candy. We actually include this data set in our revision. And this is what the sequencing image looks like. So you see an astrocyte, you see the nucleus, you see the heterochromatin region with lots of amplicons studding the interior wall of the nucleus. And these are probably the nascent mRNA, potentially non-coding RNAs, LncRNAs that are associated with the early transcription events. We're analyzing the data right now. And you have paranuclear mRNAs outside the nucleus, and they're probably also being actively translated. And then there's a big giant region where a few amplicons actually detected. And if you also look at on the right side, you see a number of empty pockets, smaller empty pockets in the cytoplasm where you don't see that many amplicons. So there's many interesting questions that rise from these images. And again, I just iterate that we have the sequencing data from these guys, bidirectional compressed sequencing, 12 bases, and we're mapping it to the human transcriptome. And then now we're just kind of analyzing the data carefully now. That, we think that this provides a path forward to looking at subcellular turgors and phase-separated granules in an unbiased manner comprehensively. And we predict that we're going to find many new interesting phenomena and features and regulatory mechanisms through our approach. So what does all this mean? So we have detected cell adhesion, channel, cytoskeletal signaling, polarity, IncRNAs, and RNA localization regulators in our manuscript. But there are a number of slides I'm not showing today because I didn't want my talk to get too long. So even though I don't have in separate slides for the points I'm making here, hopefully you'll see most of it in our revision or in the supplemented data section. So again, we have these genes from stophan to GLAG, actin, beta-actin homolog, and ZIP channel. All these genes are present in our data set. And we also know that in human astrocytes, these amplicons tend to aggregate around the rough ER region. And this is very interesting because RNA binding proteins, especially the ones that regulate translation, have been described as forming a sponge near the ER, right? So on the right, I show an example for BCOI 3' UTR and RNA binding protein. And Kristen Meyer has nicely shown in the recent years that there is a protein, RNA binding protein associated phase-separated hydrogel domain near ER that interdigitate with the ER itself, right? Exactly why, exactly how many different kinds of hydrogels are actually inside the cell, what kind of RNA binding proteins comprise them, and whether or not they actually regulate protein translation, polarity, and localization. All those things had, this subcellular interrogation of all these mechanisms had been incredibly challenging. But we think that with INSTA-seg, we now have a tool to systematically dissect these subcellular higher level RNA regulatory phenomena. So again, we're still doing analyzing our data set, but our early indication is that INSTA-seg genes and realignment patterns are similar to that of ribosome profiling, or RIBO-seg, that was pioneered by Jonathan Weissman's lab a while ago. So here, what we think is going on is that, without going through the detail, here you can see on the, I'm only going to show you the most abundant reads that we have, which is like 18S ribosomal RNA and 20S ribosomal RNA. We have mRNAs, but I'm not going to show that here for the sake of clarity. So what you have here is that in INSTA-seg, shown in so INSTA-seg is showing in a panel P, Q, and R, right? And I'm showing essentially data set, a pattern. So the read alignment pattern to the reference 18S ribosomal RNA and 28S ribosomal RNA sequence, and it's just showing you where the reads map. So poly A enriched, typical mRNA-seq, they kind of map all over the place, right? And that pattern is more or less similar whether you're looking at bulk RNA-seq or trap-seq, another way of detecting nascent proteomics, right? But in RIBO-seg, shown in K, L, and O, you have essentially can get a base pair resolution mapping of your reads to only specific locations on 18S and 20S ribosomal RNA that are exposed to your primers. And that's exactly what we get also in INSTA-seg, shown in P, Q, and R. So in fact, for 28S ribosomal RNA, the proteins are all nearly identical. So if this holds up, and again, we're doing data analysis across multiple cell types and conditions to see, in fact, whether or not, so our goal for the next couple of years is to see whether or not INSTA-seg is, in fact, closely related to the nascent proteomics, in addition to detecting non-coding RNA. And we think that the reason why reads are so heavily biased towards things that are biologically and pathway specific is because we are detecting things that are preferentially translated into proteins in vivo. So kind of going back to this earlier point that we have a cDNA stop site, right, that we can sequence, and it's part of our molecular barcode. So again, stop site and start sites, not only do they have actual information about their function, but they also serve as a UMI, a barcode, right? But for the RT stop sites, they have a basic zone incorporation. And I told you earlier that this happens across about 15% of all the reads, and the error rate for the baseline error rate internally is less than 0.1%. And if you look at the mid and lower middle graph, what you see is essentially that about 15 of these reads actually have a cis-regulatory motif that's about 10 nucleotides away from the termination site. And this is a classic feature that you would like to see if an RNA binding protein that's binding to an RNA binding protein motif is what's causing, in fact, the RT termination. So those reads where you have the cis-regulatory motif 10 nucleotides away from the air-containing RT termination site. So we have about 18 genes we identify in the Drosophila retina. And we're doing the same thing in human astrocytes right now. And interestingly, all these are, many of them are very well studied in terms of their mRNA transport, subcellular transport, and also developmental patterning, as well as cell migration and motility. And one of them that caught our eye was actin 5C, which is a homologue of human beta actin. So again, cDNA termination sites reveal potential RNA binding motifs, as I said. So on the top, you see zinc ion regulated transport related protein 48C or ZIP48C. The cDNA reads are all piled up near the poly A site. And if you look in the cDNA length is generally about 100 nucleotides. Again, there are a lot of slides I'm not showing for the sake of time. So this is a simpler condensed view of only few motifs out of the 40 that comprise a list. So this is from the data RNA binding protein database. So you have essentially multiple unique reads shown below, right? And these reads start more or less randomly, but this is all coming from the 3' UTR. But about half of the reads, they terminate exactly at the same point. Not exactly, so there's some single nucleotide variation, but the ones that have air at this termination site is shown in a yellow dot, right? So as you can tell, the vast majority of the suddenly stopping reads have these termination air misincorporation. And if you just draw a line across all the predicted motifs, you can identify several cis-regulatory motifs that are known to be involved in neural differentiation. For example, IDLAB here. And what we then do is we do an unsupervised clustering of all the predicted RNA binding protein motifs near the cDNA termination site in order to identify recurrent potentially functional RBP sites that are active in vivo, in situ, in different regions, right? So essentially this is something similar to what you would do in CLIP-seq, where you discover motifs based on our cDNA termination pattern, right, from bulk RNA sequencing data, right? And this is, in CLIP-seq, just to remind you, you have RNA binding protein specific antibody that recognizes a protein that's covalently crossing to mRNA of interest, right? So you're looking at candidate RBP interactions. Here, what we do is we're using the entire tissue as a pull-down platform, substrate, not an antibody recognizing as particular RNA binding protein. Because now we're actually pulling a large set of different RNA binding motifs and proteins, potentially proteins, and this enables us to do have less bias and do an unsupervised clustering of recurrent functional RBP sites in situ. So far I talked about the cDNA termination sites. Now I'm going to talk about cDNA start sites. Because cDNA start site starts from the polyadenylation site, So we can actually use this phenomena to look at alternative polyadenylation in situ or in vivo, right? Again, I'm going to quickly skip this slide and show that there are particular signatures associated with identifying the alternative polylation site or cleavage site. So why is this important? Well, alternative polyadenylation regulates the 3' UTR cis-regulatory element composition. So we know that 3' UTR region can be up to 50% of the entire gene, or sometimes longer, in mRNAs, highly regulated mRNAs. And 3' UTR contains multiple regulatory elements, such as microRNA binding sites, RNA protein binding sites, stem loop structures, and a number of other, and also the things that regulate subcellular localization, right? And interestingly, Kristen Meyer and a number of other people have shown that, in fact, there many genes are cleaved at the 3' UTR in a tissue-specific manner to control how long the 3' UTR is. And the shorter the 3' UTR is, you have a different set of regulatory elements that are predicted to control their biology in a different way. For example, where they're localized, how they're translated, and how they're who their protein cognate partners are. So the consensus that these events are highly tissue specific. Again, how do we study the phenomena of alternative polyadenylation in vivo that's relevant to biology, right? So what we see is that we have cell type specific alternative polyadenylation of the beta actin homolog ACT5C. And we also, so if you look at the reads, we were, again, completely in an unbiased way, we were able to, number one, identify 2 different APA isoforms of ACT5C, one with a short 3' UTR, one with a long 3' UTR. This long 3' UTR was found to have a conserved IMP or zip code binding protein consensus sequence. And we also observed that these 2 isoforms had different differential localization patterns when we looked at our physics data. And this zip code binding protein, or IMP, is an RNA binding protein known to be important for glial cell migration in flies, as well as fibrous migration. And Robert Singer has spent many years really dissecting the motif and the protein required in this particular pathway, especially for beta actin mRNA localization. So we want to have an orthogonal validation of our finding. So we, the postdoc Daniel Firth, he used a technology coming out of Arjun Raj's lab. It's called Clamfish. So, and he optimized it and he modified it a little bit so that we can actually use this to the single molecule fish. And the single molecule fish validation of these isoforms are incredibly challenging because they only differ by 400 base segment, right? Shown in this blue. Not only that, this 400 segment is also it's a low complexity sequence, meaning that it's highly AT rich, right? So doing traditional single molecule fish using tiled probes simply would not work here. And other approaches like padlock and other more sequence-specific, SNP-specific approaches also would not work here because of the sensitivity required to detect these transcripts for validation. So Daniel was able to finally optimize this clamfish for our purpose. And what we show is essentially that in the regions that start to form the optical stalk, we have a distal 3' UTR, meaning that we have the longer form of the actin 5C that's expected to bind to the Drosophila IMP, or ZIP binding protein, and it's for localizing and transporting the beta actin or Ac5C mRNA. But in the epithelial region, we predominantly see only the shorter 3' UTR. You can see these little scattered dots that shape like an epithelial cell, right? So of note that, in fact, a clownfish, and most, we find that clownfish and single molecule fish signals are super dense, so that, in fact, we had to titrate down the fish probe concentration about tenfold so that we attain about 10% detection sensitivity in order for us to quantify these dots and be sure that these are, in fact, single molecule signals, not some broad background signal. So for abundant genes, this is, I'm sure, something you guys are all aware about. For abundant genes, the sensitivity, there's a trade-off of being quantitative and specific, right? So for at least for our purpose, this worked well to validate short isoforms in situ. So so overall, INSTA-seg is a powerful tool for discovering cell type, tissue type, or location-specific regulatory mechanisms. This sets it apart from just cell typing tools. And so we are really strong on understanding the regulatory dynamics and mechanisms, molecular mechanisms, from gene transcription all the way to RNA decay, right? So we think that only by studying those mechanisms, we can actually get an insight of how cell-cell communication occurs and what they do to gene expression and how cells interpret the environment. Without that, I think we just have a lot of different cell types and microenvironmental variations without any insight into how they're actually translated into something genetically meaningful. So we have 6 images from cells and tissues. Today I showed Drosophila 3D retina, and I showed you human astrocytes. Now we're working on human breast tissue, as well as some other type of tissue sections. So now, so from 6 images, you have 12 bases. And as long as you can get reverse transcription from the 3' UTR, especially from 3' UTR, you can quickly you can go from 6 images to gene expression, RBP motifs, 3' UTR regulation, local translation, and NGS is optional if you already know the prior of how they map in the 3' UTR. If you don't have it, you do NGS as necessary for new samples. But essentially, the take home is you can do 6 imaging cycles in a single day. You can get all the mechanisms that you want out that are relevant to that many of us are interested in, which is RNA protein binding, 3' UTR splicing, and how they're translated locally in situ in single cells, right? You can get all the information out from 6 imaging cycles, we think. So much work remains, but we're very hopeful that the remaining work is more of an optimization rather than conceptual. So what are we working on? Well, we're working on ways to make the cDNA length longer, right, and more sensitive and capture more mRNA. So we're already capturing 6,000 to 10,000 mRNAs per astrocyte, but it's variable depending on who does the experiment. It's variable depending on culture conditions. So we're right now trying to develop a protocol that's much more consistent. So, and also, the cDNA length is 100 bases now, but we think that by coupling to hydrogel technology, we can make it much longer. And that would enable us to look at CMVs, RNA editing sites, splicing information on top of RBP motifs and 3' UTR regulation and translation methods. And if we can do those things, we think that INSTA-seg could really become a super powerful technology platform for studying the mechanism of RNA regulation in vivo. So our next 2-3 year goal is this. We want to comprehensively identify cis-regulatory mechanism controlling spatial dynamics of gene expression in tissue patterning. So Drosophila imaginodens is a classic primordial tissue that undergoes metamorphosis and becomes another bodily structure, right? People have been studying this for many decades, but we still only have a small glimpse of how and where these functional regulations are activated, and how the morphogen gradients, tissue mechanics, and any type of signaling events actually shape their on-off composition in individual cells across the spatial gradient, right? So what we're gonna do is we're gonna take a genetic model of Drosophila wing imaginodens, right? And there's a Drosophila line developed in the UK, and this Drosophila line has their secretion of DPP gradient. It can be perturbed using fly crosses, and when you cross these strains, what you get is essentially, here shown in the pink, you have this deep HA tag DPP secretion pattern, right? But this DPP gene can be specifically excised when you cross with another line that expresses a flippase, right, in a tissue-specific manner. So now you can actually knock out morphogen gradient in a specific tissue locations and compare the effect of gene expression, and for us, more importantly, all the cis-regulatory elements that are activated nearby, right? So you can now then have both a technical and biological control within the same tissue. And the nice thing about imaginodens is it's easily dissectable, and you can also image many multiple imaginodens simultaneously. So our goal for the next 3-4 years is to sequence about 400 imaginodens samples. And if you just we're automating this platform in collaboration with Eugene Yao at UCSD, and a fantastic student, fantastic PhD student in his lab. And we think that eventually, hopefully within a year, we can sequence 40 up to about 40 imaginodens per slide in a single sequencing run. And if you divide this up into 20 sequencing runs, we think that we might be able to actually finally understand or systematically comprehensively characterize all the cis-regulatory elements, not all of them, but certainly those that can be accessed using our technology, the current version of INSTA-se, over the comprehensively across all the regions in the imaginodens. So that will tell us how, hopefully this experiment will give us a clue, at least provide a pathway of understanding how cells determine their position in space and decide what fate they're gonna adopt going forward, depending on their location, right? So it's kind of GPS mechanism of individual cells. And more importantly, it's telling us that there's a genetic mechanism that's evolved to integrate all these signals and execute certain programs, right? This program is what we're interested in, and we think we have a way of going forward. But there are a lot of phenomena that in biology that still remains super mysterious. So this is a review from 2019. So subcellular RNA organization, a lot of people used to think of RNA, subcellular RNA, as these stringy molecules inside a bucket of water. But now there's a rush of people trying to understand, observing that RNA granules are often formed by aggregates of RNA binding proteins. And these RNA binding proteins often bind long link RNAs, mRNAs, and other small RNAs. And these things are often associated with a biological function, including splicing, translation, and such. But a lot of these phenomena have been examined in vitro in cell culture, and as you know, anytime you culture cells on glass or plastic, they already undergo a fairly remarkable phenotypic change, adaptation or change. So whether or not these phenomena actually occur in vivo, what their biological role is, and whether they're sufficient, necessary, and sufficient to induce certain biological processes or affect certain biological, none of that has been studied systematically so far. All right, we just simply don't have the method. But now our technology provides both the kind of unbiased nature of interrogation, throughput, and spatial resolution, and scalability, so that we can potentially study these RNA granules and subcellular RNA organization and their effect in local translation or RNA biology in vivo during development. And in fact, we're currently doing exactly that using human astrocyte and their migration as a model system, as a proof of concept. Finally, I want to end my presentation by talking about the Drosophila eye, complex geometry in biology, foam differential adhesion hypothesis. So we talk about cell-cell interactions all the time in the single cell biology field, right? But I advocate being a little bit more precise about what we mean by cell-cell interactions. One type that I'm very much focused on is the idea that each cell has a unique keyholes or unique affinities, so that these cells that have similar affinities or different affinities, they come together, and by through the mechanical forces, including surface tension, stress, or torsion, right? They're able to form these cell-cell patterns. These cell-cell patterns become building blocks for designing a tissue polarity or tissue growth. And then, most importantly, though, that these adhesion or differential adhesion are somehow genetically encoded, right? And this could be synaptic proteins that define neural connectome, or it could be cadherins that define cell-cell interaction in epithelial morphogenesis, epithelial cell morphogenesis, right? But whatever they are, there is a finely tuned genetic programmable network that have evolved and that lead to a precise shape that you see in biology, from spotted tigers to spotted leopard to seashells that you see on the beach, right? So understanding this genetic rule is something we're really passionate about. So I'm going to end my talk by acknowledging a fantastic postdoc in my lab, Daniel Firth. He comes from Karolinska Institute in Sweden. He's been in my lab for 2 years. He's actually a magician when it comes to computation analysis and both genetic elements and also images. But now he's becoming a guru in single molecule techniques and molecular biology. I want to also acknowledge Victor Hatini from Tufts. He was instrumental in preparing all the Drosophila eye samples for INSTA-seg, and is relevant to mechanical transduction and cell patterning in development. And the rest of the lab members from Deborah, David, Ryan, Shini, and Charlie and Colin, and also our much of our work was funded by NIH, from NIH, and with Star Consortium and V Foundation as well. Thank you very much.